This is a special episode of the Immunology Podcast, IUIS 2025, Day 4. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. Today we're back with our fourth episode from IUIS 2025. As always, we'll be discussing our favorite sessions throughout the past 24 hours of the meeting. So if you weren't able to attend, we've got you covered. We're going to kick things off in just a moment, but before we get to that,
are you tired of tedious and time-consuming cell isolation processes? Say goodbye to manual labor and hello to efficiency with RoboCep, your ultimate solution for automated cell isolation. Visit www.robocep.com to learn more about how you can reduce hands-on time, increase sample throughput, and achieve a true walkaway automation of your cell isolations.
well, want to know wasn't tiring or tedious, Brenda?
What?
My morning.
Your morning. You had a good morning?
I had a good morning of cancer immunotherapy.
Awesome. That sounds really, really fun. Why don't you share?
My cytokines were activated.
All right. Hit me with the cytokines. What's up?
All right. First in the storm of things is and both of the sessions in the morning were called cancer immunotherapy, by the way. Both actually were called cancer immunotherapy 2. One was hyphen clinical application and one did not have the hyphen.
All right. Sure.
Yeah.
That made a huge difference, I'm sure.
Yeah. Very biological nomenclature going on here.
All right. What's with cancer immunotherapy?
So the first one was very clinically oriented, which I enjoyed. The first author by Tanja, I'm going to butcher this, Jesenko from Slovenia, was about monitoring dynamics of circulating tumor cells during neoadjuvant systematic therapy in early breast cancer. So basically, she goes through a technology, lets her monitor circulating tumor cells, which is kind of cool. And then the big surprise is even early in cancer and with good clinical response, there's a lot of circulating tumor cells. Not good.
Stealth.
Yeah, they're kind of hanging out like little ninjas and they're all over the place. They don't know what this means, but it doesn't mean anything good. And they're more than previously appreciated.
Okay.
So, starting out strong with Rune in the Day. Then we had Padmani Sharma talk about checkpoint inhibitor clinical trials. And she kind of did a tour for us of where we all are at. But then she hit us with some of her own data that was just coming out in Nature Communication 2025. And that's what I'm going to talk about. So we have known that in some metastatic tumors, there's no point in cutting out the primary tumor because you're just causing injury to the patient and it's too far gone. Why bother? It's not compressing on something, right? There's no clinical reason. They actually often do worse because of the surgery, right? But no one's ever assessed this with immunotherapy. So she had a study, which is renal cell carcinoma, and one arm got surgery for either the primary tumor or a big old met, and the other didn't. And the pilots indicated survival increase with surgery only with people. You don't do surgery on people without immunotherapy, but in the immunotherapy cohort, those who got a surgery ended up doing better, which is in contrast to with other conventional chemotherapy where they did worse. So maybe time to revisit some old cancer paradigms with our new tools.
Okay.
So that was a really cool one. Then she talked about something called clonal hematopoiesis, which is generation of a very specific clone over again. I'm not quite sure why it's a new concept, but she was really talking about that when that happens, usually it leads to worse outcomes. But in checkpoint therapy, it makes things better. And then she looked at a specific mutant called TET2 and how it polarized macrophages to antigen-presenting state. And they had better outcomes in patients with this mutation in immune checkpoint therapy. So she did some nice mouse work, back to humans, back to mice, back to humans, which is really one of her points. Then we had our guest of the pod, Eric Vivier, on for NK cell therapies. And he really talked about his system the way I kind of referred to the other day about the ANKET system. So this is an antibody NK cell engager therapy. And long story short, basically these molecules use NKp46, which pulls stuff together, CD16, and then a tumor antigen, you can add a fourth one. And the whole point is get that NK cell next to the tumor so it becomes a hungry hippo.
All right.
And eats it and kills it. So then in the cancer immunotherapy part 2, as opposed to cancer therapy 2 - clinical application session. There were a whole bunch of talks here. This is like speed talks, right? Just a little too aggressively fast, but that was fine. That was not the presenter's fault. They all had like 8 minutes. So in the 8-minute speed run, One of them was looking at how the first one was called moderating regulatory T cell breaks and CAR T cell therapy of solid tumors through CD4, CD28 co-stimulation. This was by Surojit Sarkar. And he was talking about Tregs express most of the CTLA4 in the hematopoietic compartment. They're the main expressors of it. And it serves as a sink to enforce quiescence. So if you blockade the CD28, CD8086 interactions, reinforces the loss of quiescence, and then you can then use this in CAR T therapy. So if you deplete Tregs during CAR T therapy, they work better, but you have superagonism, and that's bad. And then people can die from it. Like there's too much power going on. But if you blockade the CTLA4, somehow mitigates the toxicity through this feedback loop.
Oh, okay.
So that was cool. Then this other guy named Adlai Adeshakin, also from the U.S. Again, apologies for name butchering. They basically were looking at ways to improve CAR T therapies for anti-osteosarcoma activity. And they were able to do so with a Regnase-1 knockout and B7H3 CAR T cells. Long short, it seems to be working. The effects seem to be early, and they had to modify and adjust their protocol to maintain the durable effect, but now they're going to trials because they figured out how to sort it. So that was cool. Then there was miRNA-based engineering for flexible off-the-shelf allogeneic T-cell therapy by Shah He from Singapore. Long story short, CAR T cells, sometimes they don't work as well as you want. So what they've tried to do, instead of having to tweak them all over the place and customize them, they're giving patients tacrolimus to make them a little immunosuppressed. But they make the CAR T cells tacro-resistant through genetic manipulation. So you're shutting down the host immune system and driving up the CAR T therapy at the same time. So you don't have hyperactivation of the regular immune cell from the CAR T therapy, which is a cause of toxicity, but you're having the CAR T go super boom. And then they've also found that growing cells with IL-4 and IL-7 at the same time in allogeneic T-cell allogeneic therapy in general for hematopoietic stem cell transplant reduces GVHD, and it seems to cause this by having less NK cell markers and more anti-inflammatory markers. Then there was another talk by CO Lee in Korea showing that a CTLA4, CD28 chimera potentiates CAR T cells via that CD8086 axis on both T cells and B cells. So more of the same story by other people in orthogonal labs, all kind of saying maybe we can poke on this to, the CTLA4 axis to potentiate CAR T therapies. And then there was a microbiome-derived postbiotics integrate into CAR T cell metabolism and augment anti-tumor immunity from Mike Liu in Germany. They did a big old screen of metabolites, found that pentanoate is predictive of tumor-free survival and overall survival. In contrast to other short-chain fatty acids, this paper is out on pre-release since 24, so probably stuck with a reviewer trying to get into a big journal. And that pentanoate engineered CAR T cells elicits superior tumor control, and they think it works this by pentanoate goes in citric acid cycle, our favorite, metabolic pathway of all kind. That drives the production of citrate, which they find goes in the nucleus, and they think that drives HDAC inhibition, which leads to more effector gene function.
All right. Nice story.
Yeah.
Is that it?
I am stormed out.
Is that the best you have?
That's all I have.
All right.
I did your field.
That's true. You did well.
I tried.
I'll accept it. All right. So I also have a couple of very cool stories to share. So in the morning, I've been really into the neuronal neuroimmunology, and this was a session on neuronal immune system interactions, I think it's called. And so the talks I'd like to kind of highlight are the ones from Ulrich from Andrian at Harvard University. And he talked about the pain sensation in inflammation and cancer, and the interactions between nociceptor neurons and immune cells. And I think, again, we've been talking about this for the last few years. I think it's becoming more and more clear that there are many avenues of communication between immune cells and different types of neurons. But I think particularly this TRPV1 positive nociceptors that, if I understand correctly, they are part of they are originating from this dorsal root ganglia, and they are very important for the perception of pain, of heat, of mechanical. So, but in particularly, this is discussing pain. And so there are some clinical suggestions that the perception of pain can generate inflammation. And he shows some examples of some patients that started with kind of damage to the nerves, and then they develop like dermatitis later on, suggesting that there's some kind of causality. And so he showed using a model of imiquimod-induced dermatitis, really showed that there's this close interaction between dermal dendritic cells. They sit on nerve fibers in these mice. And there is this kind of synergy between the activation of dendritic cells by imiquimod through TLR7, but also activation on a different receptor, TRPA1 on axons. And this combined activation induces production of IL-23 by dendritic cells and recruitment of inflammatory cells and kind of dermatitis. And it's really kind of this collaboration between these two signals are required for the inflammation to start. And there's this CCL2 production by nociceptive neurons attracts dendritic cells, and that's probably why they're kind of close. There is this, so that's in the steady state. You see that in situations in which you have pain, but there's no necessarily an immune stimulus. You also see that there's proteins that activate G-coupled receptors. They can generate increased production of interleukin beta, for example, on the dendritic cells in the vicinity. It's kind of priming for activation. And then if you have both things, activation of the neurons and TLR signals, there's kind of a pain and infection, then you have full-blown collaboration between this nociceptive hormone and neurons and dendritic cells. And then really, really cool also in a model of bladder carcinoma, in which there's really orthotopic, where they put they really seed the tumor in the bladder, that there's inactivation of myeloid-derived suppressor cells by the nociceptive neurons. And this promotes the bladder cancer, and this really requires this interaction between the neurons and the monocytes that become suppressors. And so, kind of continuing with the neuronal immune cell interactions, or also really cool paper from. It's a really cool story from David Heffler. And we actually have discussed this paper in our roundups, and it's really cool. It's the finding that there are normal brains have T cells, right? That they find in normal cerebrospinal fluid and in brain of mice. They find that there's resident CD4 cells that are expressing interferon gamma. They're interferon gamma in normal CSF. And this is kind of, we always think that this is not good to have these inflammatory things. Is usually we associate that is not, but that's what they see. And there are cells that are in the parenchyma of the brain, so they're inside the brain tissue. And it's particularly in this organ called the subfornical organ, and that's really where you see this presence of T cells that they express CXCR3 and TBED. They will produce interferon gamma, and there to some extent they have some similarities with T cells found in the white adipose tissue. And I guess it makes sense because there's a lot of lipids in the brain, like so maybe that's why. And so these cells appear around, I think this says 4 weeks of age in the mice, and it requires it kind of depends on the presence of microbiota. So germ-free animals have less of these CD4 cells in the brain and also the white adipose tissue. And if you already colonization with these short filamentous bacteria already restores this CD4 development. So there's something they are doing that it's important for their development. So there's this, it looks like they are really migrating from the gut to the brain or the white adipose tissue. And they show this with photoconversion experiments. And it's funny because they also analyze cerebrospinal fluid from donors, including David himself. So he analyzed his own cerebrospinal fluid to look for these CD4 cells, and you also find them in humans. And there's kind of, a lot of similarities in the phenotype of these cells. If you have, so one of the things is CXCR6, you see them in this CD4 cells on the CSF. And in CXCR6 deficient mice, you have lower numbers of CD4 cells in the brain, but not in the adipose tissue. And this is kind of a key difference between these two cell populations. And it looks like the cells have a kind of function, or they have an impact, because interferon gamma knockout mice have different, some small but apparently significant differences in some of their behavior when it comes to anxiety, and tests called the novelty suppressed feeding behavior, in which the mice are more likely to go feed somewhere new, or they get more anxious and so they stop feeding because they're not feeling comfortable. And I think also that was a story they already published, and they have this new story about the about Parkinson's disease, and how Parkinson's, you might be able to really classify Parkinson's and autoimmune disease originating in the gut. Because they coming back to these T cells in the CNS, there is a period in Parkinson's, in early Parkinson's, and probably before kind of formal diagnosis of Parkinson's, that is called the prodromal Parkinson's, in which people already have this very specific disruption of the sleep that almost invariantly will correlate with development of full-blown Parkinson's. So similar to multiple sclerosis, there seems to be stages of kind of immune activity in the brain. And in patients that have this early stage prodromal Parkinson's, they see increased inflammatory cells in the CSF, increase of leukocytes in the CSF of these patients. And he was saying something along the lines, and this, when they take the CSF, they just measure, stuff in the serum, and then they throw away the cells, and the cells might have all the answers we're looking for. There's an increase in microglial-like macrophages in the CSF, and at this stage, and increased T cells as well. There are some, they measure some of the kind of the gene networks, increase of TNF alpha, JAK-STAT, and MHC class 2 genes are upregulated in Parkinson's disease mouse models, and also in the CSF of CFS of prodromal PD patients. And this, in a way, they think that there's this initial kind of inflammatory response, and then the neural kind of the neuronal degeneration comes later as a second phase. And so that if you could pick up at this initial stage, and you could block, for example, I think they said with Humira, that's a TNF blocking antibody, right? I think they suggested that maybe if you treat the inflammation early on, you could reduce the onset or kind of delay the onset of the actual, Parkinson's. And so they're going to get a lot of money to make a clinical trial for that. So they're getting started for that. So that sounds pretty exciting. And then a couple of afterwards, it was abstract talks from Treg topics. And yeah, I like the talk from Jeanne Verrieken from the University of Hasselt in Belgium. And she looked at the importance of regulatory T cells in brain barrier integrity, and showed that there's this subset of IL-34 producing regulatory T cells by the blood-brain barrier that protects their permeability. And if you remove, and that if you have like less IL-34 in serum is something that is observed in this EAE model in mice, so of brain inflammation, and there's kind of this initial reduction of CD34, late reductions in IL-34, and this correlates with more permeability. And this is also something that you also see in patients that they have lower IL-34 in early stages, because it has already been seen that there's an increased permeability of the blood-brain barrier that is higher at the beginning of the onset of this disease. So basically, it looks like IL-34 production from Tregs protects this integrity of this blood-brain barrier. It was a very short talk, so I guess there's probably more to talk about this. But yeah, it's nice to see that the Tregs just keeping things real. And yeah, so I think these are the talks I'd like to highlight for today. It was really fun.
Some neuroimmunology. Is there going to be a third postdoc for you, Brenda?
A new life. I mean, it is really cool, especially, the implications are profound.
Yeah. I was back when I was about to become faculty, starting on some neuro gut processes where we were going to put in transwells epithelial cells on a 2D layer. So you likely do this air-liquid interface, but on the other part of the well that was connected, you're going to have brain organoids in 3D matrigel and then link them with media through the fluidic chamber that was there and see the brain talking to the gut and back. Nice.
Yeah, I mean, there clearly is some kind of ongoing conversation between them. So
the gut has I forget the number, but has quite a number of nerves compared to the brain.
Yeah. It's
the neurons, in terms of neurons in the gut versus the brain. It's something way closer than you would think.
Yeah, I also remember that being kind of impressed. I don't remember the numbers, but I was thinking it's not like orders of magnitude less. It's
very close. All right. Well, you know what? If one of our guests remembers that number off the top of their head, or listeners rather, remembers that number off the top of our head, they can tweet it at us at @immunopodcast or on Blue Sky at immunologypodcast.com.
Indeed.
And you know what? That does really bring us to the end of our show.
Awesome.
Here. So check back tomorrow for another episode recapping the final day of the meeting. See you then.